In this first activity, we will examine the external features of a monocot leaf. Monocots are one of the two major groups of flowering plants, and their leaves show a set of consistent structural patterns that make them easy to recognize once you know what to look for. As you work through this activity, you will focus on two main features: the patterns of veins in the leaf and the way the leaf attaches to the stem. These characteristics reflect both how monocot leaves develop and how they function mechanically and physiologically. Monocot leaves have several external features that distinguish them from eudicot leaves. They are typically long and narrow, with veins that run side by side from the base of the leaf to the tip. This pattern is called parallel venation, meaning the veins do not form a branching network. This parallel venation supports the even distribution of nutrients and water along the length of the leaf. It also provides structural reinforcement for leaves that often grow vertically. Most monocot leaves are also sessile, meaning they do not have a petiole. Instead, the base of the leaf forms a sheath that wraps around the stem. The point where the leaf attaches to the stem is called a node, and the region between two nodes is called an internode. As you examine your specimen, pay close attention to the direction and arrangement of the veins and how the base of the leaf connects to and surrounds the stem. When we think of monocot leaves, we often picture narrow grass blades with obvious parallel veins and a sheath wrapped around the stem. But many monocots look very different at first glance, which can make these features harder to recognize. Let's look at a few examples. Banana leaves are extremely broad, so the veins may appear complex. However, if you follow the secondary veins, you'll see that they run in parallel from the central midrib toward the leaf margin. At the base, each leaf forms a large sheath that tightly overlaps the previous one, creating what looks like a trunk, but is actually a stack of leaf bases called a pseudostem. Palm leaves may be fan-shaped or feather-shaped, which can disguise their venation. Even so, each segment of the leaf contains veins that run in parallel from base to tip, and the massive petiole still arises from a base at the stem. Orchid leaves are thick and leathery, often strap-shaped. Their veins run lengthwise and parallel, and the leaf base clasps or partially wraps the stem, showing the same monocot attachment pattern. Ginger plants have broad leaves that look almost eudicot-like, but their venation remains parallel, and their stems are actually formed from overlapping sheathing leaf bases, just like in bananas. Even though these monocots vary greatly in size and shape, they all share the same fundamental structural plan: parallel vascular bundles running the length of the leaf, and a leaf base that forms a sheath around the stem or pseudostem. This reinforces an important idea in plant anatomy. Surface appearance can vary widely, but internal developmental patterns, like venation and attachment, reveal true evolutionary relationships. Before making your observations, pause and consider this question: why do monocot leaves typically have parallel venation, and how does this relate to their overall structure? Consider the shape of the leaf, the way it grows, and how veins must support and supply water to the blade. Write your prediction in your lab notebook, then continue. Here we have St. Augustinegrass, a common warm-season turf grass in Florida. Leaves arise from distinct nodes along thick, creeping stolons, above-ground horizontal stems, with internodes between them. Record the name of the specimen that you will sketch later. Now examine both the upper and lower surfaces of the leaf. Ask yourself, are the veins more pronounced on one side of the leaf? What is the overall vein pattern? Are the leaves aligned in the same direction? Do you see any branching, or do the veins remain parallel from base to tip? Monocot leaves exhibit parallel venation, in which multiple veins enter the leaf at the base and extend toward the tip without forming a branching network. This arrangement provides efficient transport and mechanical support along the length of the narrow blade. Next, observe how the leaf attaches to the stem. Determine whether the leaf is sessile, meaning it lacks a petiole and attaches directly to the stem. Look for a sheathing leaf base that wraps around the stem and helps anchor the leaf in place. Identify the node where the leaf emerges and note the internode region below it. Now you will create a labeled sketch of the St. Augustinegrass leaf. In your lab notebook, draw the overall shape of the leaf. Clearly show the parallel venation. Label the sheathing leaf base and at least one vein. Focus on accuracy of structure rather than artistic detail. Pause the video while you complete your drawing, then continue when you're finished. To summarize this activity, monocot leaves are characterized by parallel venation, with veins running side by side from base to tip, a long, narrow blade shape, and a sessile attachment with a sheathing leaf base that wraps around the stem. These features reflect how monocot leaves grow and how their vascular system is organized. Once again, we see the central theme of plant anatomy. Structure supports function. In the next activity, you will compare these features to those of eudicot leaves and see how different developmental patterns lead to different leaf architectures.